thesis. The title of the talk is Regulation of Sleeping Beauty Transposase, which is a DNA transposon, and how it is regulated by host-encoded cellular factors. And this work was done under the mentorship of Dr. Shushna Ishwa from the Laboratory of Mobile DNA, Max Delbruck Center for Molecular Medicine, Berlin. So what are transposable elements? We all know our genomes are littered with a mobile genetic elements called transposable elements. It can jump from one place to another, from one place of the genome to other place of the genome. It was first discovered by a scientist called Barbara McClintock in the year 1994. And she was a plant genetist, and you can see here is the kernels, is a corn kernels. And she is very much interested to know why there is a difference in the color in the corn kernels. And she wants to understand the molecular mechanism of it. So what she understood is that even though all this kernels is coming from the same cell, the difference arises because there is something in the genome that jumps from one place to another place. And from that, she discovered the transposable elements. The discovery of transposable elements changed our understanding about genome. Genomes are not static, they are susceptible to change. And this transposable elements, just because it can jump from one place to another place, it can function as an insertional mutagens. And finally, the transposons, they can also regulate the nearby genes. As I said, our genome is littered with the transposable elements. And this transposable elements can jump from one place to another. And finally, for the discovery of transposable element, she was awarded the Nobel Prize in the year 1983. So this just gives you an overall understanding of transposable elements. As I said, the transposable elements is widespread across many organisms. You can see it is present in fish, Tanio, in the fly, in humans, in rat, bat, and even in the plants. And there are three types of transposable elements. One is called LTR transposons, non-LTR transposons, and DNA transposons. And there are other transposons like helicates. So in our talk, we are going to concentrate on DNA transposons. We are not going to concentrate on RNA transposons. We are going to concentrate on DNA transposons. And this slide just gives you a summary that the transposons are widespread across many organisms. So today we're going to talk about DNA transposons. How does a DNA transposon looks like? You can see it has two terminal repeat separated by a gene called transposase. So the transposase is an enzyme which acts on the DNA ends, cut them, and insert them into some target location. So today we are going to talk about a transposon called Sleeping Beauty transposon, which belongs to the TC1 mariner. And what is a TC1 mariner family? It is the most widespread DNA transposon which is found in eukaryotes. It ranges from plants to humans. And what happens is when a transposon enters into an organism, during subsequent course of time, it accumulates mutation. And through mutation, it loses its jumping ability. And that is what we call it by vertical inactivation. And what my PhD mentor, Dr. Shushna Ishwa, did is she activated this transposon. What she did is the transposons, which has undergone vertical inactivation, she corrected those mutations and resurrected them. Resurrected means what? Gave back the life. And she checked whether this transposons can jump. And fortunately, yes. And that is the reason she named the transposon as a Sleeping Beauty transposon. A Sleeping Beauty transposon has characterized by two terminal inverted repeats, and in the center, it codes for a gene called transposase. And Sleeping Beauty was the first DNA transposon to be shown active in vertebrate cell. I repeat, Sleeping Beauty is the first DNA transposon to be shown in activity in a vertebrate cell. And it gave us an opportunity. The first opportunity is if at all a DNA transposon is active inside the cell, how it is regulated. And second one is it has a widespread application. First is it can be used for transgenesis. That is, you can put a foreign gene into a vertebrate cell. And we are going to see how it can be applied for gene therapy applications. So, as I said that a transposon is characterized by a terminal repeat and a central gene which codes for a transposase. What happens is when the transposon gets activated, it started coding for an enzyme called transposase which binds on the terminal repeats. And then what happens is it brings the terminal repeats together, and basically it looks for a TA dinucleotide in the genome where it goes and integrates the transposon. And as a result of its jumping into the new location, what happens is the target site undergo duplication, TAT. And we found many host factors which plays a very important role in the regulation of Sleeping Beauty transposon. One such host factor is called HMG2-LAC1, which acts on the UTR region, which also functions as a promoter sequence, and thereby increasing the transcription of transposase gene. And we also found a DNA bending protein called HMGB1. And we also found out that when the DNA transposon is moved from one location to another location, it is reported by a host factor called Cu70. And we found another host factor called NIS1, which plays a very important role in the regulation of DNA transposon. That is, it basically regulates the cell cycle of the cell. So, how does the host factor HMG2-LAC1 regulate Sleeping Beauty transcription? As I said that the DNA transposon of a Sleeping Beauty has two terminal repeats, and the terminal repeats on the left we call it as 5' UTR, on the right we call it as 3' UTR. And we found that this HMG2-LAC1 binds on the 5' UTR and drives the expression of transposase. And what happens is when the transposase is produced in a high quantity, in a sufficient quantity, what happens is there is a feedback inhibition. It goes back and binds on the HMG2-LAC1 and downregulates its expression. So what is this HMG2-LAC1 protein is like? HMG2-LAC1 belongs to a high mobility FOX protein, and it plays an important role in Wnt signaling by downregulating it. And other than that, there is not much information available about the protein. So what is the objective of my PhD work is to understand how this HMG2-LAC1 regulates Sleeping Beauty transposon and whether this interaction is conserved in development. And since HMG2-LAC1 is a human protein and Sleeping Beauty is a fish protein, we want to understand the evolutionary conservation, whether it is conserved across different species. So, the first and foremost, what we did is we want to understand the physical interaction of HMG2-LAC1 with Sleeping Beauty transposon. So, what we did is we took the HMG2-LAC1 and Sleeping Beauty gene construct and expressed in a HeLa cells. When you express, the proteins are produced. And through an experiment called co-immunoprecipitation, we want to check whether there is a physical interaction or not. First and foremost, what you see in the term, we want to check by Western blotting whether the proteins are expressed. So the proteins are expressed. You can see the antibodies against the protein. This is a Western blot. So we check the proteins are expressed, and then we did the protein pulling assay, in which we confirmed that when we pulled HMG2 like one, which is tagged with a HA, we saw the Sleeping Beauty coming. So this kind of confirmed the interaction of HMG2 like one with Sleeping Beauty protein. So we want to understand the molecular interaction of the HMG2 like one protein in relation to Sleeping Beauty degradation. So what we did is we did a yeast-2 hybrid system. So this yeast-2 hybrid system, what you can do is you can screen a library for your selective protein. In this case, we selected HMG2 like one and we screened for the proteins. What are the interacting partners? We found out that HMG2 like one interacts with two different proteins called SUMO1 and PIAS1. And what is the SUMO1 is? A SUMO is a post-translational protein addition to any protein. It is a member of a SUMO protein family. And PIAS1 acts as a ligase. It enhances the SUMOylation of a protein. So, as I said, what is a SUMO? The SUMO is a post-translational addition to any protein. So, it is always present in an inactive form. It gets activated by three steps. First is the activating state, where the GG tail of it is cleaved. And then it is conjugated. Finally, the E3 ligase, which I said is a PIAS, plays a very important role in targeting the activated SUMO1 protein to its target protein. So this plays an important role in the attachment of the SUMO1 protein to its target protein. So when the SUMO is added to any protein, what happens is it changes the functionality of the protein. And this whole process is a reversible in nature. That is, it can be reversed. The SUMO can be attached and detached, attached and detached. So this is all about SUMOylation. So when a SUMO is added to a protein, what else? How does it change the functionality of the protein? We can see that it can change the protein-protein interaction. It can change the subcellular nucleation, transcription, DNA binding, and also it changes the higher order chromatin structure. So the first and foremost, we want to check whether HMG2 like one can be modified by SUMO or not, whether HMG2 like one when it is reacting with a SUMO or not. So what we did is, again, we transfected HMG2 like one protein along with the SUMO1 protein in a HeLa cells, and what you see is a Western blot against the HMG2 like one protein. And when a SUMO protein is added, what happens is it increases the molecular weight. So you can see when it is not added, you can only see HMG2 like one, but when the SUMO protein is added to the HMG2 like one, you can see a slow migrating band. And this slow migrating band indicates that HMG2 like one is modified by SUMO. And SUMO exists in a cell in three forms, SUMO 1, 2, 3. And what we found out is HMG2 like one can be modified by all three forms of SUMO proteins. And further, we also confirm by pull down assay, where we expressed SUMO as well as HMG2 like one and pulled the HMG2 like one. And you can see the clear bands, which indicate slow migrating bands, which indicates that HMG2 like one is modified by SUMO 1. And it is modified at two places. Another thing what we want to find out is whether this whole process is irreversible in nature. So as I said, the SNP protein acts as a SNP protein acts as a protease, which makes this SUMOylation process irreversible. So what we do is We did the same simulation assay in presence of a SNP protein, and this is a wild type SNP1 protein. This is a mutant, and what we found is when we added the wild type SNP protein, it does reverse the process. So this kind of confirms that the simulation of HMG2 Lycopene is reversible in nature. And we want to know whether this bias protein, which is also one of the protein that came out in the these two hybrid system, and we want to check whether this bias protein is involved in the simulation of HMG2 Lycopene. Again, what we did is we did this simulation assay. What you see is a Western blot picture of the HMG2 Lycopene protein, and we overexpressed PAS1 protein. And what we found out is the addition of PAS1 doesn't change the simulation of HMG2 Lycopene. That means it doesn't cause any addictive effect. So without PAS1, whatever the simulation of HMG2 Lycopene is there, it is there. And further, I said this is just a comparison of the proteins of human HMG2 Lycopene with many other HMG2 Lycopene proteins of different species. And we found out that the lysine of K317, this is the position of the lysine, K317 and 320 is conserved across species. And what we did is we made the lysine to get converted into arginine so that it is not recognized by the simulation machinery. And we carried out a same simulation assay, and you can see in the presence of sumo, when you change this lysine to arginine, it is no more simulated. And this we call it as a mutant HMG2 Lycopene. And we want to check what is the effect of simulated HMG2 Lycopene on Hsp transcription. We said the sleeping beauty transcription plays a, that is HMG2 Lycopene enhances the sleeping beauty transcription. And we want to check what is the effect of SUMO1 on the sleeping beauty transcription. So what we did is we took the 5' nuclear of sleeping beauty transposon and fused it to a luciferase gene. So this luciferase gene is a reporter gene which can give the activity of the 5' nuclear promoter. And what we found is in the luciferase assay, very interestingly, when you add HMG2 Lycopene, the transcription of the UTR goes very high, but in the presence of sleeping beauty transposon, the transcription goes down. That is, when the HMG2 Lycopene is modified by SUMO1, the transcription goes down. And this is statistically relevant. Whereas is very interestingly, what we found out is the mutant HMG2 Lycopene, which cannot be modified by SUMO1, the transcription is similar. That means the mutants doesn't affect the transcription of the transcription function of the HMG2 Lycopene, but it can no more be modified by the SUMO1 protein. So, and also we checked it by a colony assay, and we also got a very similar. This is a colony assay. Sorry, unfortunately, I couldn't go back to the previous slide. So this is a colony assay, which kind of indicates that the same. This is a colony assay where you transfect the cells and you put them under antibiotic selection, and you will find out the number of colonies. And similarly, the wild type HMG2 Lycopene, it showed a good amount of colonies. But when we add the SUMO modifies the HMG2 Lycopene, and the colony number goes down. Whereas the mutant, they have a similar kind of a colony number. So the decreased transposition activity of SBP in the presence of SUMO. And what is its relationship of HMG to Lycopene with the SB transposon in development? So, see, a transposon is a selfish DNA parasite. For it to be very successful, it has to enter into the germ cells and early part of the development, so that it can, first, if it enters into the germ cell, it can go to the next generation. And in the early part of development, it can get transposed to all the cells. So, this is one of the reasons the transposons, they target the germline events and the early development events. So, since since sleeping beauty transposon is a fish protein, what we want to know the expression of the HMG2 Lycopene. HMG2 Lycopene, as I said, is very conserved across organisms. We want to check its expression in a zebrafish, which is very easily Danieri, which is where you can easily obtain all the different stages of the embryonic development. So first we looked at the mRNA expression of HMG2 Lycopene. This is a fish HMG2 Lycopene in different stages. So it is nicely expressed in the zygote, cleavage, blastula, gastrula segment. So till blastula it is expressed. After gastrula, it goes down. From gastrula to hatching, the level goes down. And what this diagram indicates is HMG2 Lycopene is nicely expressed in the early developmental stages of a organism. And we also want to check the expression in the germ cells. So in our lab, we developed a way to develop the to culture the germ cells. So in this case, we use the rat germ cells, and you can also differentiate this stem cells. So what we used as a example of a germ cell is spermatogonial stem cells. So the first is we want to check the molecular marker. That is, whether it is a germ cell. So we use FGFR3 for the expression. And then we check the expression of HMG2 Lycopene protein. And we found out the HMG2 Lycopene protein is nicely expressed in the spermatogonial stem cell, which is a model for germ cell. And we also checked at the protein. So the protein is nicely expressed when it is germ cell. When it is differentiated, the level goes down. And finally, we want to check the assay to link whether the sleeping beauty transposon can be nicely expressed in the germ cells. So this is an assay we developed. And this is a PCR-based assay. We want to check whether HMG2 Lycopene, sleeping beauty transposon can be expressed inside nicely expressed, and the results of which I'm not discussing. This is an assay we developed, general assay we check. And what is the Ebenov means? The Ebenov keeps the protein inside the cell. So we want to check the evolutionary conservation of this interaction of HMG2 Lycopene with sleeping beauty. And what you see is a phylogenetic tree of HMG2 Lycopene. So HMG2 Lycopene is spread across the different species, like humans. It is present in rat. It is present in fish. Everything. And do human and fish HMG2 Lycopene. We also clone the fish HMG2 Lycopene, and we want to check whether it can drive does the same promoter activity of sleeping beauty. And what we checked is the luciferase assay, which we express the reporter gene, and it is nicely driving the expression of the promoter, whereas the truncated one, we remove the part where it binds. When we remove, you don't see the promoter activity. And this is a expression of the fish HMG2 like one, and you can see it is nicely expressed. And very interestingly, it also can get modified by SUMO1. And we also check the luciferase assay as well as the colony assay. And very interestingly, what we found out is here, when we compared with a human and the fish, the fish HMG2 like one has much more better activity when compared with the human. And the same results we observed in the colony assay. And also, what we found out is that the fish protein can also be modified by the SUMO1, and it can downregulate its activity. So just to give a summary of this part of my work, this loop, the SUMOylation of HMG2 like one downregulates sleeping beauty transcription. And we can see that it is expressed, the expression of this fish HMG2 like one is there in the early development as well as in the germ cell. And the function of this HMG2 like one is highly conserved across species, and its interaction with sleeping beauty is also conserved. And now I'm going to talk about the application of DNA transposons for transgenesis and gene therapy application, and how it can be used for targeted gene exchange in plants. So there is a hyperactivity of sleeping beauty that is available, and it was chosen as the molecule of the year in the year 2009. And it has a widespread application. It can be used for generating stable cells. It can be used for modifying the genome of many vertebrate species like cow, pig, goat, etc. And it can be used for gene therapy applications. So what is this application project of Sleeping Beauty is? The Sleeping Beauty can be made as a two-component vector system where you can remove the middle gene and you can replace it with a gene of interest, and you can have a transposis. And then what we want to do is we want to first modify the genome of a rat with a green fluorescent protein, and we want to change the green fluorescent protein with a genome of our interest. And this is combining the Sleeping Beauty transposon technology along with the Cree technology. And what we want to do is what is the advantage of this is, first thing is we want to select the safe locus. Safe locus is the one in which the genome, that particular genome, is free of any disturbance. It doesn't have any NHs, it doesn't have any kind of a disturbance from other part. And this kind of a technology is never demonstrated in rat, because rat is used as a model animals for many diseases. And first, using this technology, we created a green fluorescent rat combining the Cree-LOX technology along with the transposon. We created a green rat. And then what we did is we took out the embryo of the green rat and using the Cree recombinase. This Cree recombinase, what it does is it can exchange between the two LOXP sites. And we want to exchange this green rat with a red. We want to exchange the red gene with a green gene with a red gene, and we managed to exchange it. So such a kind of a technology was first demonstrated in the rat. And this is further confirmed by the PCR that the exchange is not a random integration and it is a targeted integration. And this is an example of how Sleeping Beauty can be used for genome modification application. So in this project, we demonstrated a single copy of the transposon can be integrated into a region of a rat, and then it can be changed in a targeted manner. And this will pay for developing a humanized rat models, which has a widespread application in medical field. And I would like to thank all the members of my team, where I carried out my work, and especially Dr. Shushna's work, who's wearing a white coat, and now also these are the members of the team. So thank you very much for your attention, and if you have any questions, you can ask me. Thank you.
I am Dr. T.N. Uma Maheshwari, working as Professor and Head of Admin in the Department of Oral Medicine and Radiology, Savita Dental College and Hospitals. I would like to use this platform to share my research experience of formulating various targeted herbal drug formulations for management of oral potentially malignant disorders. To know what is oral potentially malignant disorders, they are red and white lesions, suspicious lesions occurring in the oral mucosa before the development of oral cancer, and mostly they are associated with the use of tobacco, either in the form of smoking or smokeless by the patients. To define it, they are also called as epithelial precursor lesions with altered epithelium having increased likelihood for progression to squamous cell carcinoma. Globally, the prevalence of oral potentially malignant disorder ranges to about 1 to 5%, and in India, it is slightly higher in the range of 13.7%. Individually, the most prevalent oral potentially malignant disorders are oral submucous fibrosis, with a prevalence percentage of 6.42%, leukoplakia, which is a white patch, which ranges around 5.2%, and erosive lichen planus, which is an immunologically mediated disease, which also ranges in the range of 2 to 2.6%. What is most important is the malignant transformation rate of these oral potentially malignant disorders, which accounts for about 2.2% global level. And in India, it is even more slightly higher in the range of 17%. Individually, if you see, oral submucous fibrosis is having a higher malignant transformation rate as 13%, followed by leukoplakia, oral lichen planus. Any oral potentially malignant disorder with a histopathological report of mild or moderate or severe dysplasia, their chances of malignant transformation rate is going to be even more higher in the range of 36.4%. This is the reason I had chosen oral potentially malignant disorder instead of choosing oral cancer as my research thrust area, and I am working on formulating a lot of drugs since there are no exclusive local drug delivery for management and preventing this malignant transformation of oral potentially malignant disorder into developing into squamous cell carcinoma. Hence, I am concentrating on chemo prevention. In the year 2015 to 2018, I had involved in formulating a muco-adhesive buccal film for management of these potentially malignant disorders with the aim of preventing malignant transformation using lycopene, curcumin, and beta-carotene. Why have I chosen this muco-adhesive film in order to ensure targeted drug delivery is because it is a bi-layered film technology with active drug in the muco-adhesive layer. There is a backing layer which will facilitate unidirectional flow of drug. It adheres very well to the oral mucosa and completely dissolves within 10 to minutes. There are no adverse effects, and as well as there's no altered taste sensation noted by the patients. It ensures rapid drug absorption and is designed to optimize delivery across the mucosa. Before entering into this buccal adhesive film formulation, I involved in doing a systematic review to review the literature, to analyze the literature, what are the different topical therapeutics which are available in managing these oral potentially malignant disorders so far done by the various researchers. And I found, after searching PubMed, Cochrane, Central Library, and Science Direct Research, as well as Google Scholar, there were around 89 articles, and then after proper screening, it was 33. And After analyzing each of these 33 articles, I found that there were only 16 clinical trials who have tried in formulating these drug formulations in management of oral potentially malignant disorders. I also did a quality assessment of these 16 studies using a RevMan software, and I found that few studies had high risk of bias, which you can see has red markings, and few studies were having moderate risk of bias, but many studies were done with a proper study protocol and they were considered as low risk of bias. To summarize the results of the systematic review conducted by me, I found that there were a lot of cases which has been concentrated in lichen planus and leukoplakia followed by oral submucous fibrosis, and most of the researchers have formulated a gel in managing these oral potentially malignant disorders, and many of them have concentrated in using the Arovit or the vitamin A in the form of T-retinoin. And regarding the outcome, many of the patients had a lot of improvement in burning sensation. Before and after, the VAS score or visual analog scale depicted that there was a significant improvement after using these drugs. Mouth opening, which is a main problem in oral submucous fibrosis, also had significant result, as well as cheek flexibility, followed by tongue protrusion. So there were 16 clinical trials so far done in various topical therapeutic formulations in oral potentially malignant disorders in varied population. Around 6 clinical trials were conducted in India. And total number of cases included so far by the earlier researchers were around 692 cases, in which most of the cases were lichen planus, leukoplakia, and oral submucous fibrosis were little less in number because the prevalence of oral submucous fibrosis is more in India. And out of all the 16 clinical trials, one clinical trial involving raspberry gel had a very proper study protocol with proper 6 months follow-up and 90% of complete response. So the lacunae which I identified in the existing literature is that exclusive local drug formulation for oral potentially malignant disorder is a need of the hour, and we need to combine the different antioxidants in order to get a better result. With this lacunae identified in the previous literature search, my research question for the current research proposal was whether lycopene, beta-carotene, curcumin combination as a muco-adhesive film is effective in treatment of oral potentially malignant disorder. The rationale behind this study is because most of the oral potentially malignant disorder, around 65%, definitely turn into oral squamous cell carcinoma in the later run. And correct diagnosis and early treatment can definitely prevent this malignant transformation. Exclusive topical therapeutic for chemo prevention of oral potentially malignant disorder is a need of the hour. Hence, this research was conducted with the aim to evaluate the effectiveness of lycopene, beta-carotene, and curcumin combination as a muco-adhesive film in treatment of oral potentially malignant disorder, with various primary and secondary objectives in order to evaluate the reduction of burning sensation, whether there is any reduction in the size of the lesion after using this muco-adhesive film, and how easy it is for the patients to apply, and whether any adverse effects were noted, as well as the main thrust area of mine, chemo prevention, is observed or not. So the study design was an interventional clinical trial. Patients who were clinically and histopathologically diagnosed to have as oral potentially malignant disorder was included, with proper institutional ethical human clearance was obtained before conducting the clinical trial. The sampling was judgmental sampling because it was not a randomization. Those patients who were clinically diagnosed as well as histopathologically diagnosed as oral potentially malignant disorder, such as leukoplakia, oral submucous fibrosis, and erosive lichen planus, which is considered as the most prevalent oral potentially malignant disorder, was included, and the study was conducted in Savita Dental College. The main inclusion criteria are any oral potentially malignant disorder which was graded as mild dysplasia. When it is graded as moderate or severe dysplasia, it is not suitable for medical management. It is suitable for surgical management. So those cases were not included in the study, and patients who are willing to undergo histopathological examination was included. patients who are already under treatment were not included in the study. To know what is oral submucous fibrosis, what is most important to understand is that it is a disease which is characterized by fibrosis of the oral mucosa, which occurs because of usage of areca nut, which contains a lot of alkaloids. Pan, Hans, Mava, these type of smokeless tobacco or chewing tobacco is associated with the formation of oral submucous fibrosis. As you could see in the picture, the patients will have inability to open the mouth will occur very stiff, and they also will have severe burning sensation. And when they are associated with other oral potentially malignant disorders, leukoplakia, their malignant transformation rate is going to be even more higher. Similarly, there is a disease called leukoplakia, which you could see it as a raised, elevated, non-scrapable white patch. This is of smaller in size, and as the lesion increases, the size of the lesion will increase, and it will also be spread out throughout the buccal mucosa. And sometimes this white patch will develop even a redness, which is a sign that it is turning into malignant transformation. So these are the various stages of leukoplakia, and leukoplakia is a white patch, a suspicious white patch, oral potentially malignant disorder, and is usually associated with smoking habit. Another disease called Lichen Blindness, which occurs in the skin, and for some patients it occurs both in skin and oral mucosa. For some patients only in the oral mucosa as an area of desquamation of the gingiva, as well as in the periphery you can see radiating white lines, which is called as Wickham's striae, a clinical presentation, a classical clinical presentation, which can be easily diagnosed that it is a case of Lichen Blindness. It's an immunologically mediated disease, but few cases of erosive Lichen Blindness is associated with dysplastic change in histopathological examination, and they are more prone for development of oral squamous cell carcinoma in the later run. So in all these 30 patients, what was assessed is the visual analog scale. The burning sensation was numerical rating scale done by the patient, and then the inter-incisal distance or the mouth opening was measured. The size of the white patch was also measured, and histopathological examination grading of dysplasia was measured before starting the clinical trial. The proforma or the data extraction collected from all the patients include all the demographic data, as well as the type of tobacco, the frequency and the duration of usage of either the smoking or smokeless tobacco, the white patch or the restricted mouth opening, the fibrosis of the oral mucosa in oral submucous fibrosis. All those clinical details were completely recorded. The size of the lesion was measured both on the right and left buccal mucosa, the anterior posterior extent, the superior inferior, the area of the lesion, VAS score before starting the treatment, and mouth opening was also recorded. The patient was recalled every 15 days once, and the muco-adhesive films were supplied to the patients at every visit. The patient was called at every 15 days in order to record again the size of the lesion, area of the lesion, as well as the VAS score and mouth opening in each of the visits, so that to see whether there is any improvement. Similarly, if the patient has noticed any discomfort such as dysphagia, burning sensation, ulceration, altered taste sensation after using this muco-adhesive film, that was also recorded, and those patients were discontinued. And the easy of application, and or there is any other discomfort apart from what it has been noted in this proforma, was also recorded and was clearly elicited from the patient. Now moving on to the preparation of this muco-adhesive film, the main components were beta-carotene, lycopene, curcumin. The other components which helped in formulating the films were methoxy, ethoxy, polyvinyl pyrrolidone, carbopol, sodium alginate, carbovax, ethanol, and distilled water. The polymeric solution was prepared by ethyl cellulose dissolved in 10 ml ethanol, then hydroxymethyl cellulose was added to the alcoholic solution, stirred very well for about 30 minutes. The sonicated solution is to get a clear solution to mix it with propylene glycol, which is a plasticizer. And finally, the drug was dispersed in 5 ml of ethanol sonicated for 10 minutes. The drug solution added to polymeric solution under continuous stirring, and it was finally poured into a plastic Petri dish. Again, was dried at 40 degrees Celsius. It was cut and packed in an aluminum foil, as you could see, like this. in a circular format. The drug components are 1:2:2 lycopene, beta carotene, and curcumin combinations. These are the following physiochemical evaluations such as film thickness, drug content, determination of weight uniformity, surface pH, fold endurance were all checked. Even the mucus adhesion, because it's a mucus adhesive film, the mucus adhesion, the muco-adhesive residence period was very well observed before applying it to the patients. And these are the demographic data of all the patients collected for the clinical trial. Especially the tobacco history was very important, and we had recorded, as well as the type of lesion, whether it is lichen planus or leukoplakia or oral submucous fibrosis, was clearly recorded. And it was found that most of the patients were oral submucous fibrosis, because as I told earlier, the prevalence of oral submucous fibrosis in Indian population is slightly higher. And most of them had tobacco habit, and few of them had tobacco and alcohol habit. 36% were the patients of lichen planus, which is not associated with habit, but it's an immunologically mediated disease, which is also considered as a potentially malignant disorder. And we had made sure that all the oral potentially malignant disorder was subjected to histopathological examination to know whether there is dysplasia or mild or moderate or severe dysplasia, because only no dysplasia and mild dysplasia was included, so that we can ensure chemo prevention for these type of disorders. This is how the film was applied, and was instructed to the patients also how they have to apply at home. And this is how the film starts disintegrating in few minutes and finally gets dispersed entirely to the lesion. Post-assessment also, the visual analog scale, the burning sensation, mouth opening, size of the lesion was recorded. As I told earlier, for every 15 days, the patient will be recalled, and all the pre-assessment and post-assessment parameters were completely documented. The outcome parameters mainly we concentrated in the clinical reduction in the size and the symptoms also, such as burning sensation, the severity of the lesions, mainly the dysplastic stages was also recorded. The burning sensation between the baseline and at the end of third month had significant statistical improvement, as well as the size of the lesion also. At every visit, there was definitely a reduction in the size of the lesion. Mouth opening had even more statistical significant improvement when compared to VAS score, as well as the size of the lesion. These are the few images of pre and post clinical assessment of lichen planus cases, and you could make out the size reduction of the lesion clearly. This was between the baseline and at the end of the treatment. The mouth opening has significantly increased from, it was around 28 and it was increased to 34 millimeter in one patient. And you could also see the vascularity of the lesion, the mucosa, it was totally blanched or white in color before the treatment. After the treatment, there was increased vascularity, and the normal suppleness and the normal pink color mucosa is you can appreciate it after the treatment. So based on this muco-adhesive film formulation, what was next and how are we going to improvise this muco-adhesive film was that going on in my mind. And finally, I had now involved in herbal drug in situ gel formulations using these various other herbal drugs such as raspberry, tulasi, aloe vera, and black turmeric. And this was tulasi, aloe vera, turmeric in situ gel. Earlier, I was formulating a muco-adhesive film. Now, I'm involved in formulation of in situ gel. 1 gram of tulasi, aloe vera, turmeric mixed with 100 ml of distilled water, boiled for 15 minutes, and again filtered and concentrated till 10 ml. The antioxidant activity of this herbal formulation was assessed using DPPH assay. Antimicrobial activity was also assessed in various species like Candida, Streptococcus mutans, Lactobacillus, Staphylococcus aureus, because these are all the oral flora. And the antioxidant property also showed significant result at 50 microliters of concentration. And the antimicrobial activity also was good for Streptococcus mutans. Another in situ gel formulation which we are now currently working is Lycopene raspberry formulation. Lycopene percentage is around 10%, raspberry 25%, and there's this preparation is also subjected to various testing like anti-inflammatory testing using bovine serum albumin, which showed inhibition percentage of 90.2% in 10 microliter concentration itself, cytotoxic activity using brine shrimp lethality assay, as well as spectrophotometric reading of antioxidant assay done by DPP HSA revealed that 50 microliter of extracts have a relatively high absorption dosage with inhibition percentage of 61.3. Antimicrobial testing by agar diffusion method reveals maximum area of inhibition around 16 millimeter against Streptococcus mutans. So all these properties have been assessed before we are going to try it on human clinical trials. So I thank my mentor, Dr. Gnanasundaram, who has always been a backbone for all the academic activities involved by me. My own university, which provides a very good infrastructure to do such research work, which is going to be really beneficial for patients, rather than working on oral cancer. Working on oral potentially malignant disorder, which will develop into oral cancer, is very important. So full resource of patients as well as infrastructure enriched Savita University. I'm very thankful for always being a support for us to do such research. I thank the organizing team of 41 IABMS annual conference organizing team for giving a wonderful opportunity to share my research experience on chemo prevention of oral potentially malignant disorder. Thank you, one and all.
Saraf, Untitled Medical College, also practicing Siddha system of medicine more than 15 years. The main causes is excessive heat. Heat usually occupational and tightness of wearing inner gears. The lorry driver, bus driver, sitting and driving more than 8 hours, 10 hours. So that causes excessive heat producing in gluteal region. It affects that scrotum and defect in spermatogenesis. And another one is drug. Drug, long-standing steroidal, any hormonal like this affected in the spermatogenesis. It causes further male infertility. And toxicants. That's toxicants usually the pesticides. It causes for also male infertility, stress, work stress, and environmental and society, family stress usually affect that spermatogenesis. Excessive exercise. Usually the bicycling, example. Of long-standing exercise and bicycling is causes for the it affect blood flow to the scrotum, so it causes chronic disease like cancer, diabetic. Diabetic complication usually diabetic retinopathy, the vessels vascular affected. That is also causes for the spermatogenesis. The inflow of the blood flow to the scrotum also decreased during the high diabetic disease. And dietary deficiency, it is also causes for vitamin deficiencies. The varicocele also one of the major cause. And then genital tract infection, undescended testis, obesity, genetic cause. This is further causes for male infertility. Next. Evaluation and diagnosis. How to evaluate and diagnose the male infertility. The semen analysis. The seminal analysis usually quantity is 1.5 ml to 5 ml. And seminal quality also need for the conceive of the sperm density. Normal density is usually more than 15 million per ml. Total sperm motility more than 40%. And sperm morphology also major causes. That is the normal percentage sperm is lower is not more lower than 4%. Infection also, usually we are seeing this analysis. WBC is very high, it's causes for infection or inflammation in our body. Other variables also, level of zinc, citric acid, and phosphate, alpha glucoside also there. We can rule out that during the normal evaluation sperm analysis. Next. WHO reference stage, we are everything we are majorly referred to WHO only. Usually total sperm count per ejaculation 39 million to 928 million. Ejaculate volume 1.5 to 7.6 ml. Sperm concentration 15 million to 250 million per ml. The motility 40 to 80%. The progressive and non-progressive both. Usually progressive motility should be 32 into 75%. That is the must for the good quality of the sperm. Next. Anti-sperm antibody test, hormonal analysis, follicle stimulating hormone, luteinizing hormone, thyroid stimulating hormone, testosterone, prolactin also need for the evaluation for male infertility. Genetic test if severe or needed for if severe oligospermia. Less than 5 million, we have to analyze for that genetic evaluation test. Other kind of study is imaging study, that is scan, transrectal, USG, scrotal ultrasonography, also helps to rule out the male infertility. Next. I am simply give this very few points about the causes and how to evaluate the male infertility, because my topic is how to treatment aspect male infertility. So I am concentrating about it. Siddha system of medicine. That is main aspect in Siddha aspect. Andathil ullade pindam, pindathil ullade andam. That is what is in environmental. It is also reflect to our body. The environmental made up of 5 elements. The same constituents also contains in our body. So there is no different from space ingredients and our body constituents. That is our one of the basic principles of our system. Next. First, our Siddha system of medicines, more than 4,000 years ago BC, the system was transmitted by Siddhas, Siddhi attaining perfection. We are not going to see surgical correction and primary control and idiopathic Saturn. Next. Infertility type and treatment. Defective spermatogenesis is oligospermia, oligoasthenospermia. Most of the cases responded oligospermia. Oligospermia is a decreased count of sperm. We are treating those kind of patients. It's very good result during and after the treatment period. Azoospermia, it is very difficult to treat. It depends 50% possibility. We are going to treating 3 to 4 patients. For example, 2 patients will success and 2 patients couldn't success. Necrospermia remarkable changes after treatment. We can combinedly treat with allopathic medication. Most kind of medicines available for male infertility in our Siddha system of medicine. We are going to see one by one. Next. First step is oil bath. The beneficiary of oil bath, hormone promoter, muscle relaxant, increasing blood circulation, balancing Tridosam. Tridosam means Vada, Pitta, Kapha. In Tamil, Vadam, Pittam, Kapha. Hormones increasing blood circulation. It is a high volume of blood circulation enter into the scrotal. It is also facilitating for spermatogenesis. For oil bath, Chukutailam usually in Vada dosam and Kuparienne and Chandranadi Tailam also used for Pitta dosam. Chukutailam and Lemanakatailam used for Kaba dosam. Sperm motility defect occur in Vata dosam. Decreased sperm come out occurs in Pitta dosam. Structural defect, necrotic sperm occurs in Kaba dosam. Next. The Vada dosam, we are using the Nahaparpam. It's a highly based preparation, 100 milligram along with milk twice a day. Ayachanduram also we can take. It is the iron ingredient based 100 milligram with honey 2 times per day. For Pitta dosam defective patients, we are using Linga Chanduram 50 milligram along with adjuvant honey. Silasathapadpam also used 100 milligram with 10 gram of Ventusine legium. Kaba dosam Rasagandhi Viligu along with or palm jangri. Rasagandhi Viligu dose is 50 milligram to 100 milligram depending upon the patient's weight. Tangaprapam also used to especially Kaba dosam and the 3 dosam defective spermatogenesis 100 milligram along with 2 gram Ashwagandha plain powder. Not only used to the spermatogenesis defect, also Rasaparpam also used. It is mercury based. Tangaprapam and Rasaparpam both are utilized for 3 doses of spermatogenesis defect. Next. Thermal factor. For excessive heat, we are treating for the patients following medications. Silasathapadpam 100 milligram with the milk, Ventusine legium 10 gram twice a day. Lichen legium also used for this thermal factor, that is reducing the heat of the body 10 milligram per day, 2 times per day. Sadaviri also used 5 gram 2 times per day. Chitramutti Madakatailam, that is the major role to reducing the thermal factor, 10 drops with milk. Next. Varicocele and obstructive factor. Agaprapam used for the obstructive 10 milligram per day. Ayachanduram also used for the varicocele and obstructive factor of the male infertility. Kalachichuranam utilized internally and externally both. And also for other external treatment, Dapat we are using the bitumen or Kalachichurnam, applying with equine, pork or paneer water externally around the scrotal during the night times. Next. For infectious disease, following the medication for utilization, one capsule that is 500 milligram dosage. Also, 1 milligram, 100 milligram, Nandi Miliku 1 capsule, Tankaparpum 50 milligram, Serangotaini 5 drops with the milk. As above said, adjuvant. Next. During the chemotherapy or radiotherapy also causes for major role in male infertility. For that condition, we are using Lycopene 10 milligram 2 times per day. Also, used Tetramethyl 10 gram per day. And Tripla Karpamatre 2 tablets 2 times per day. Sinthil Churnam 1 gram 2 times per day. Ajimadra Churnam 1 gram 2 times per day. We are using as said above adjuvant milk like that. Next. The environmental, usually the pesticide and stress, we are using the Aspanda powder, proper oil bath, Murgi Pulegium, Bremine, Sadaverine, Mutupur. We are using the stress and strain and pesticide causes of male infertility. Next. Endocrine factor. There is the major role of male infertility. That is Northern 10 gram. We are using for this endocrine factors. Udubulegium. This is the system called Jiva Sarak. Udumbulegium. 10 gram with milk and also Tangaparpam. 50 milligram along with milk. It is a very high success rate of male infertility. Next. This is the yoga therapy. Surya Namaskaram. It contains 12 steps. Padmasanam, lotus position. Bhakti Maustasanam, Bhujangasanam, Sarvangasanam, Halasanam, Dhanurasanam, Kapurasanam. It is asanam is for helps to achieve therapy of male infertility. That benefit is next. Next. Yoga Mudra also a major role as in the previous one. Yoga Mudra, Daspini Mudra, Varun Mudra, Vajrali Mudra, Linga Mudra, and O Mudra. It is used for the male infertility. Mudras. Next. External therapy, full body Tokkanam and Sirodhara Nasiyam used for during treatment of male infertility. Benefits, facilitate blood flow to lower abdomen and testicle to promote spermatogenesis. Next. The diet usually take patients to usual diet. Ghee, milk, sugarcane juice, garlic, pepper, dry fruits like almond, dates, walnut, plus vegetables, sandal powder, fresh juice, and lot of veggies. And green leaves, Murugai kire, that is Moringa olifera, Arakire, Amaranthus tricolor, கருந்தொலைக்கீரை அப்போமியா manchega, பசலைக்கீரை Islamica. It is have high potent spermatogenesis activity. So usually we advise to take weekly, twice or thrice alternative days. Above set reach to the patient. Next. Other than commonly used the polyherbal combination, many more than 50 polyherbal combination available in Siddha system of medicine. I am mentioning few herbal combinations only, that like Nirjil Pidai Churnam, Oril Tamare Churnam, Elepanekilang Churnam, Punekali Churnam, Karundutti Veer Churnam, Tamare Magaranda Churnam powder, Sub Churnam. Not only this, it's limitedly many polyherbal combination available to treat male infertility. Next. If you have any query, I am very happy. Thank you.
Thank you one and all, and special thanks to the organizers of this wonderful CME for giving me an opportunity to present before you. My topic of presentation is about the metallotherapeutic approach of Siddha medicine containing lithium as an antipsychotic drug of choice. We all know that microelements and trace elements play a crucial role in cell functions at biological, chemical, and molecular levels. There are other group called functional trace elements, which are getting attention from a research and medicine perspectives for its explicit role in treating specific conditions. Examples of functional trace elements include lithium, boron, silicon, strontium. Out of the various functional trace elements, lithium is a popular drug of choice for bipolar disorder, that is manic depressive illness. It has a well-known established pharmacology as an antioxidant, immunomodulatory, signaling, anti-apoptotic, and neuroprotective effects. One of the drawbacks and pitfall of lithium therapy is its narrow therapeutic index, that is more chance of having adverse side effects to the patients. Siddha medicine also utilizes a n number of raw drugs from natural mineral resources for preparing numerous potent formulations, and we are having a systematic approach of material processing, manufacture that ensures safe, bioavailable, target-specific and potent drugs. The drugs which are prepared from mineral origin is a good source of macro, micro trace and functional elements, but it is still unexplored and documented. Numerous salts, metals, mineral compounds or subminerals are utilized in Siddha medicine, and the final products of such wonderful formulations are called purpum, identically called as oxides, chenduram, sulfides, churnam coincidence oxides, padangam sublimates, dravagam distillates. Understanding the main lithium source as marine products. So our aim was to identify Siddha medicines that is prepared from marine products, especially in the form of calcination oxides or calcoxides, and to sort out Siddha medicines particularly used for psychiatric illness. After that, we estimated the lithium content of these marine calcine formulations to substantiate its therapeutic possibility in treating psychiatric illness. So we selected 7 out of important Siddha formulations in calcine form prepared from marine products, and we analyzed each sample through ICP-OS for its elemental composition of lithium. And for our understanding, we come to an observation that one of the medicine called Muthuparpam had a significant level of lithium in it. In Siddha medicine, pearl is a highly valued raw material credited with numerous therapeutic properties. And there is a special indication noted in the Tamil manuscript verses of Siddha medicine about a word called Dhairyanattam. It symbolizes depression, solitary attitude, lack of confidence, excess fear, and lack of concentration, which is treated efficiently with the pearl oyster. The nanocalcined oxide form of pearl, otherwise called Muthu Parpam, has been widely used to improve cognitive functions as a nerve tonic for neurological deficits, depression, psychiatric illness, delirium, epilepsy, and is more common in Siddha clinical practice. Understanding the traditional view blended with the scientific approach, we come to a conclusion that Muthu Paspam has the ability to act on brain, especially as per the Siddha medicine. It has special activity in Chandra Mandala, that is zone of intellect, consciousness, creativity, and calmness. And when we characterize Muthu Parpam, it is alkaline in nature, and the presence of lithium and its pharmacology in neurological pathways substantiate its use in psychiatric illness, because lithium has multiple level of action in brain structure, neurotransmitter modulation, intracellular changes, etc. It has inhibitory action on dopamine receptors and glutamate receptors, and has an excitatory action and stimulatory action on GABA receptors. Lithium combination, despite its role in treating psychiatric illness, various side effects are reported that are of particular concern to clinicians. So the search for a natural remedy that can substantiate lithium drugs is in high demand, which points to traditional systems like Siddha medicine. The presence of lithium content in the Muthu Parpam may substantiate its use as an antipsychotic drug. Muthu Parpam may be promoted as a safe and effective substitute for lithium drugs. Further clinical researches are appreciated to validate the efficacy of the drug. Thank you.
Hello everyone. Topic of my research study is the cytotoxic and apoptotic activity of Cannabilia seed extracts in HT29 human colon carcinoma cells. The extracts which were used are lectin-rich extracts. So what are lectins? Lectins are carbohydrate binding proteins which are widely used in glycobiology. The concannavalins, they are mannose glucose specific lectins from a leguminous plant Cannabilia. Among concannavalin A, in short con A, from Cannabilia ensiformis is widely explored for its structure and functional properties. Con A is widely used mitogen. It's also used in cancer research. There are also other concannavalins reported, such as con G from Cannabilia gladiata, con C from Cannabilia catatica, con M from Cannabilia rosea. So for the present study, we used con A, con C, con M, con G, and compared their cytotoxic and apoptotic activity in HT29 cells. The seed extracts were prepared. First, the seeds were collected from all 4 accessions of Cannabilia, and it was homogenized in sodium chloride. The homogenate was further precipitated by ammonium sulfate at different saturation levels, and for each in each step of fractional precipitation, protein was estimated and specific activity was determined by hemagglutination assay, and the fraction with the higher specific activity was dialyzed and used for cytotoxic and apoptotic assays. The cytotoxicity of seed extract, concannavalin extracts were determined by MTT assay, and apoptotic activity was determined by morphological observation using acridine orange and Ethereum bromide dual staining and flow cytometric assay. and its effect on apoptotic regulating genes like P53, AKT1, ERK1, and 2 was determined by quantitative reverse transcription polymerase chain reaction. Coming to the results, this is a result for MTT assay where concannavalin seed extracts, induced dose-dependent decrease in viability of cancer cells. The IC50 value obtained from this graph was depicted in the column table here, where you can see Con-G showed higher activity compared to Con-C and Con-N. So hence Con-E and Con-G extracts were further explored for their apoptotic potential. The IC50 and half IC50 and quarter IC50 values were used for AOEB staining. The morphological features like the chromatin condensation, nuclear fragmentation, apoptotic body was observed, which indicates apoptosis. As a result for flow cytometric assay, where the proliferation phases like G0, G1S, G2M were decreased, and there was a substantial increase in sub G0, G1 level, which indicates apoptotic cells. So the results of flow cytometry is in correlation with morphological analysis. Based on the cytotoxicity and apoptotic assays, Con-G was further studied for its effect on apoptosis inducing genes like AKT1, ERK1, ERK2, and P53. And the QRT-PCR results showed Con-G can induce downregulation of all these tested genes. So in the present study, the concannavalin extracts showed selective cytotoxicity in HT29 cells. The difference in binding affinity is due to the altered glycosylation pattern on the cancer cell surface. Con-E and Con-G induced apoptosis as observed in morphological observation by AOEB staining and accumulation of sub G0, G1 proportion in flow cytometric assay, which holds good with previous reports. The apoptosis regulating pathway genes were downregulated in this study, and the observation is supported by published literature. So in conclusion, in this study, we report that the Con-G has a potential lectin for sugar specificity, cytotoxicity, and apoptotic efficiency. So here are some of the references. Thank you.
Good morning to all. My topic is about pharmacovigilance study on Lichen material, mineral formations prescribed in NIS. My abstract ID is 195. Introduction. The system is most ancient system of which concentrate on medicinal plants and spiritual disciplines. It is broadly classified into three types. They are Tavaram, like herbal products, Tavadu, ignoring exceptions, and Jivam, animal products. Siddha system gives more importance to the use of herbals and minerals for treating the diseases. Karpu Vishnu Chakramatri is one of the herbo-mineral preparations indicated for Janni, Andavadam, Timirvadam, Ilamilevadam, Parisavadam, Tanurvadam, Penisam, Sevirokam, Valipugal, Suram, Irumal, Kunmam, and Kuttam. As per ancient Siddha literature, the drug used for treating diseases should undergo proper purification and processing, and also it should be taken with suitable adjuvant, accurate dose, proper administration regime, As quoted in classical text, the causes of adverse drug reactions are improper purification of drug, overdose, unsuitable adjuvant, drug interaction, and drug intolerance, drug allergy, inappropriate diet regimen. My aim to the study adverse drug effect of airborne mineral formulations Karku among OP and IP patients in Ayurveda's hospital. Objective to document the drug effect of OPD and IPD patients treated with Karku to analyze the characteristics of adverse effect along with herbal combo medications. Study type is observational study, study design, cross-section study period 6 months, sample size 200 patients, studies place Ayurveda's hospital, National Institute of Subject selections. I will inform to all departments about my study. I requested to refer the patients reporting with adverse of Karku. Inclusion criteria: age 18 to 60, both 6, both OPD and APD patients in Ayurveda's hospitals. Exclusion criteria: patients who were under the allopathic medications or any other alternative medications, patients with any history of any other illness, any drug allergy. Questionnaire. I take this type of questionnaire. And he developed symptoms like gastritis, abdominal pain, and oral ulcer. In the study, how 5 out of 200, like 2.5% patients developed ADR. 2.5% adverse drug reaction like gastritis, abdominal pain, and oral ulcer at the time of administration of third week. None of the ADR was fatal out of total ADR. Commonly reported ADR was gastritis, abdominal pain, and oral ulcer. Summary: as per Hansen's drug, the drug used in treating and all proper administrations and diet regime as quoted in classical cases. The causes of adverse drug reactions are improper purification of drug, overdose, unsuitable adjuvant, and drug interaction, drug intolerance, and drug allergy, inappropriate diet regimens. Karfu is one of the herbal preparations indicated for Janni, Andavadam, Timirvadam, Tilambilayvadam, Parisavadam, Tanurvadam, Penisam, Sivirogam, Palipugal, Suram, Irmal, Kunnam, Erikanum, Kuttum. The aim of the study study the drug effect of herbal formulation of Karfu among OPI and IP patients in Ayurvedic hospital. The literature about Karfu has been studied in this study. 5 out of 200, 2.5% patients developed in ADR, 2.5% adverse drug reactions like gastritis, abdominal pain, and oral ulcer at the time of administration of ADR. Conclusions: the incidence of rate of ADR is found to be much lower, 2.5% than reported. None of the ADR was fatal out of total ADR. commonly reported earlier was gastritis, abdominal and oral ulcer. Furthermore, research is needed to better understand the underlying reasons for the difference in area. Thank you.
Hello, warm welcome to all. Myself, Dr. S. Rajamani, PG Scholar, Department of Gunapadam, National Institute of Chitta, Chennai 47. My topic is effectiveness of Sindal Churnam on Valial Keelvayu, rheumatoid arthritis. My abstract ID is 209. Introduction. Rheumatoid arthritis is very complicated autoimmune disease in worldwide. It affects the women than the male. It aggravates in winter season. It affects the joints. Symptoms like morning stiffness, swelling of the joints, restricted movements of the joint, difficulty to handling the object. In many of the Siddha literature, the Sindel is the one of the immunomodulatory effect. So I select the Sindel Churnam. Is one of among the mentioned in classical Siddha literature for the treatment of various Vata disease, skin disease, diabetes mellitus, and urinary tract infection. It is also known as it is also known for its anti-inflammatory and analgesic properties. Aim to assess the clinical efficacy of Sindel Churnam on Valial Kilvayu. Methodology, it is an experimental study type. Study design, open clinical study. Sample size 20 patient of both sex. Sampling method, purposive sampling. Study places in our OPD, Ayodhya Pandita Hospital, National Institute of Siddha, Chennai 47. Study period is 6 months. Sindel Churnam ingredients: Sindel Kodi, Karisale, purified Punahams, earthworm. Dosage 1 to 2 gram. Adjuvant is key. Duration twice daily for 2 months. This is the results in blood parameters levels: hemoglobin, white blood cells, red blood cells, eosinophil, in mean, standard deviation, and p-values. Seems like a blood glucose level, urea level, and RA factor. Results: the swelling measurements is noted in above one before and after treatment. In knee circumference, 3 measurements: right upper, middle, and lower, on right side and left side. This is the before and after. This is the ESR value in before and after treatment. This is the swelling measurements on right and left side. RA factor in before and after treatment we were noted. This is one of the pain scale using the arthritis universal pain scale in before and after treatment we were noted in this study. Results: clinical symptoms before and after were noted. It showed significant clinical improvement. Reduced the swelling measurements and in universal pain scale also. The study drug Sindel Surnam has 70% of significant results and 30% of shows moderate results on Valial Kilvayu. Biostatistical study, since the p-value is significant in most of the clinical manifestation among those patients for the treatment of Valial Kilvayu. Discussion: among the 20 patients, all were improved both subjectively and objectively. Clinical symptoms before and after were noted. Thus, the clinical trial study showed significant clinical improvement in certain manifestation of rheumatoid arthritis, like major and minor joint pain, morning stiffness, swelling, restricted movements, and reduced the universal pain scale measurements. Conclusion: the trial medicine Sindel Churnam showed good results with relieving symptoms in almost 70% of patients, and it is concluded that the treatment was effective and significant. Once again, Siddha medicine is proved itself as a great boon to mankind. As this is a hospital-based study, the trial conducted in smaller population and efficacy of Sindel Churnam among those population is found to be potent. The further clinical trial to be done in larger number of samples to confirm the efficacy of the Sindel Churnam. Thank you for the organizing committee for giving the opportunity. Thank you so much.
Greeting to all of you. I am Dr. Devanji Swaran, PG Scholar, Department of Konabadam, National Institute of Siddha. Today I am going to present a topic, comparative study on qualitative and quantitative analysis of Inji Chorasam and Inji Charu prepared as per Siddha literature. Abstract ID 211. Introduction. As reviewing the Siddha literature, all formulations are giving specific vehicle in accordance with the disease condition of the body. Some of the herbal drugs which are used as vehicle may be processed before administration. Among these, the Inji Charu is commonly used as vehicle. Inji Charu has more volatile oil which are produce pungent effect. So the process of Inji Chorasam will reduce the concentration of volatile oil and mitigate the pungent effect. So far is no scientific study with regards to the Inji Chorasam. The study is selected to compare the qualitative and quantitative parameter of Inji Chorasam and Inji Charu. Objective: preparation of the Inji Chorasam. Next, to evaluate qualitative and quantitative analysis of the Inji Charu and Inji Chorasam. Study methodology. First, prepared method of sample A, Inji Charu, ginger juice. Collected gingers were measured weight and peel the outer layer of ginger. Peel gingers was weighted. Gingers was crushed on stone mortar to get the juice. The juice was kept for a few minutes to sediment. After that, the juice was filtered and measured the volume. Then the juice was divided into two equal parts. One half of the juice was stored in a airtight glass container named as A. This is sample A. Next, prepared method sample B, Inji Chorasam or ginger decant. The mud pot was heated until the source sound. Here is sprinkled a few drops of water in it. Then second half of the juice was poured into the heated mud pot. After the juice was filtered and measured the volume, finally the juice was stored in an airtight glass container named as B. This is sample B. Samples were analyzed by following instruments: HPTLC, FTIR, and GCMS. Result and discussion. This is histogram under the HPTLC scanner capture at 450 nanometer and 250 nanometer light. Here, when the red line cut the red squares, that's are proved as presence of same type of component in both samples. According to the histogram at 450 nanometer light, 12 number of component present in Inji Charu and 10 number of component present in Inji Chorasam, but 10 number of same component present in both samples. According to the histogram 250 nanometer light, 10 number of component present in Inji Charu and 9 number of component present in Inji Chorasam, but 7 number of same component present in both samples. Compare the functional components of Inji Charu and Inji Chorasam using FTIR analysis. Through the FTIR analysis, alkaline ester nitrosulfonate, sulfonic acid, sulfoxide, and sulfoaminate functional components are present only in Inji Chorasam. GCMS chromatograph data interruption of Inji Charu and Inji Chorasam. In Inji Charu, through the analysis, 4 components are found in Inji Charu. 32.67 area percentage is noted as a higher percentage. That is chemical name is oxandidic enonic acid for hydraulic methyl ester. Through the GCMS analysis, 5 components found in Inji Chorasam, 39 point area percentage is noted.
as a higher percentage. That chemical name is oxygeninic 9-enonic acid. Compare the functional component of ginger juice or injicharu and gingerdican or injicharism using GCMS analysis. Pentadienionic acid, methyl ester, and 10-octadienionic acid, methyl ester component.
I'm going to present my research topic entitled on the ascendancy of betel leaf in the metabolism of trophoblast. Pregnancy is a physiological state to sustain developing fetus accompanied with high energy demand and an increased oxygen requirement. Providing a balanced nutrition to the developing fetus during normal pregnancy is associated with immense stress along with metabolic changes and vascular remodeling. During preeclampsia, there is reduced trophoblast invasion and inappropriate spiral artery remodeling occurs with elevated blood pressure resulting in placental stress affecting the development of fetus and the current world is mainly in search of new molecule of herbal origin, and there is increased incidence of drug toxicity of synthetic drugs, and natural compounds are easily available. They are cost effective and having less or no side effect and does not require any special method of preparation. And betel leaf is selected for this study because it is rich in flavonoids, phenols, saponins, quinones, tannins and tritapenoids. Most of these compounds are antioxidant and antihypertensive in nature. Hence, this can able to manage the maternal as well as fetal complication like hypertension and fetal growth restriction resulting in low birth weight babies. The objective of the present study is to isolate the placental trophoblast and subjecting them to in vitro hypoxia in a dry gas incubator, and the isolated trophoblasts were incubated with two different extracts of betel leaf. One is salivary extract, other one is diastase extract. Why salivary extract is preferred for this study is that it is rich in enzymes such as diastase, lipase, protease, DNase, RNase, and it is also having antimicrobial, antiviral property due to the presence of lactoperoxidase and lactoferrin. And the present study aimed to ascertain the vasoprotective effect of betel leaf because it is anti-inflammatory, anti-hypertensive, antioxidant, and it can able to prevent the placental platelet aggregation. So all these property can be effective in the management of maternal hypertension and able to supply sufficient nutrient to the proper growth of fetus. Hence, the present study was performed or carried out to illuminate the efficacy of salivary as well as diastase extract of betel leaf with the concentration of 1 milligram and 1 hour incubation. Two different subjects were selected. One is normal, other one is pre-eclamptic plasmal trophoblast, which is characterized with proteinuria and high blood pressure. These are the various methodologies follows, and the statistical analysis were performed by student t-test, and difference in the results were expressed with the difference in the p-value less than 0.01, p-value less than 0.01 and p-value less than 0.001. Three different parameters were assessed. One is leptin, second one is cytochrome 19A1, the third one is adiponectin. And while talking about cytochrome 19A1, it is an enzyme involved in the synthesis of beta estradiol. It has got its effect on trophoblast differentiation, proper perfusion of placenta, vascularization, and it is able to decrease the systemic vascular resistance, and it can increase the blood flow. And alteration in the metabolism and energy balance between the maternal and fetal interface. Also, the pathophysiological events during placenta were assessed by these three parameters. Talking about leptin, leptin plays a crucial role in the energy intake, utilization, and energy homeostasis. This 16 kilodalton protein hormone called as leptin plays an important role in the proper placental trophoblast and invasion, as well as it is also involved in various biological aspects such as inflammation, immune response, reproduction, and angiogenesis. During normal pregnancy, leptin is produced by human placenta tissue and has a vital role in the regulation of food intake and energy expenditure through the homogeneous expression of its receptor in the extravillous trophoblast. Next one is adiponectin. Adiponectin has a role in the formation of steroids and cellular cell survival, vasodilation, oxidation of free fatty acids, and cytoproduction. All these are important in maintaining the energy homeostasis. Thereby, it can prevent the low birth weight babies and premature babies. Adiponectin is a 30 kilodalton protein secreted by both maternal as well as placental adipocyte, involved in the regulation of fatty acid metabolism, cell proliferation, and differentiation. Adiponectin plays a crucial role in the energy metabolism and adaptation of materno-fetal interface via regulating the glucose utilization, lipid synthesis, energy homeostasis, and vasoregulation. Next one is cytochrome 19A1. This aromatase enzyme also plays an important role in preventing the premature baby and in maintain the normal growth and development of the fetus. While these three levels were assessed, leptin, adiponectin, and cytochrome 19A1 were assessed in normal trophoblast as well as pre-eclamptic trophoblast, there is a significant increase in the level of leptin and decrease in the level of adiponectin as well as cytochrome 19A1. When trophoblast was subjected to in vitro hypoxia, there is increase in the level of leptin and decrease in the level of adiponectin and cytochrome 19A1. And interestingly, it was noted that when normal placental trophoblast were subjected to in vitro hypoxia, the level of leptin, adiponectin, and CYP19A1 somewhat resembles the placental trophoblast, which are collected from pre-eclamptic subject. And when these trophoblasts were subjected to diastase as well as salivary extract of betel leaf, there is a decrease in the level. There is a decrease in the level of leptin, and there is increase in the level of adiponectin, and there is increase in the level of CYP19A1. And when subjecting the trophoblast, we are pre-incubating the trophoblast with salivary as well as diastase extract of betel leaf and then subjecting them to in vitro hypoxia, there is an increase in the level of leptin and there is a decrease in the level of adiponectin and decrease in the level of CYP19A1. So the differential expression of leptin, adiponectin, CYP19A1 influences the energy imbalance with response to external stimulus such as low oxygen tension. As well as oxidation process. The ascendancy of betel leaf extract on the metabolism of trophoblast require sufficient oxygen tension. It is inferred from the in vitro hypoxia study by subjecting both the trophoblast to in vitro hypoxia and incubating them to salivary as well as diastase extract of betel leaf, where it is unable to modulate the level of all these three parameters, and it is indicating that oxygen plays a crucial role in the proper development of fetus. The future aspect of this study is the interaction between the bioactive compounds of
A very good morning to respected dignitaries. This is Vishwohini, research scholar pursuing PhD in biochemistry in the Department of Biochemistry, Bharathiar University, Coimbatore, under the guidance and leadership of Dr. E. Padmini ma'am. I'm here going to present a piece of my research work entitled In vitro cell cytotoxicity studies of a polyherbal extracts in vivo E6 cell lines against dengue types. Aimed to explore the medicinal plants in the place of synthetic infections, things like dengue. Therefore, on the present study dengue fever. Hexane, ethyl acetate, acetone, ethanol, and aqueous were selected for the present study as solvents. Cold percolation method was adopted and the extracts were screened already for the presence of phytochemicals and for further quantified for polyphenolics. As we all know, polyphenols are known immunoregulators. For cell viability test was done for the extracts by adopting MTT assay, and the cell lines chosen for the study is being Vero E6. Further, ethanol extract of the said medicinal plants were characterized and the optimum bioactive compounds were performed for in silico analysis using iGen.2. Subculturing was done in Vero E6 cell lines, and the cells were treated with trypsin phosphate virgin glucose. Trypsin will act on cells, cleaves the protein component, loosens its morphology, and detaches the protein content. Virgin ETTA is used as a chelating agent. Phosphate buffer is meant for pH maintenance, and glucose will have its nutrient supply. The adate cells are washed with phosphate buffer, and the washed out cells are mixed with minimum essential medium without fetal calf serum. 100 microliters of cultured cells are loaded in a 9-6 well plate and are then observed in a compound microscope to identify the morphology of the cells. The cells were then incubated at 37 degrees Celsius for 2 days. In order to have viral isolation procedure from the sample, it is further carried out where the confluent cells were tested for the absence of contamination. Growth medium was decanted with TPCK trypsin without fetal calf serum. Media was pipetted and 200 microliter of sample was inoculated in a T25 flask for 30 minutes at 37 degrees Celsius. 2 micrograms per ml of TPCK trypsin was later added and incubated at 37 degrees Celsius in 5% carbon dioxide incubator. Flasks were then observed for cytopathic effect. It means the virus has to go and invade the Vero cell line and it has to disrupt its normal morphology. When there is a 75 to 100% CPE observation was done, the flask contents were harvested by collecting the supernatant fluid and added with 0.5% bovine serum albumin as a stabilizing agent. Even when there is no CPE, after a week's time, the cells has to be harvested. To confirm the presence of virus in the sample, the following tests were done. Number 1 was a RNA extraction by adopting a conventional PCR, where denaturation, annealing, and subsequent template extension was carried out at the mentioned temperature and time limits. Further, heme agglutination test was performed in order to detect the presence of virus in the sample tested. For this, geese RBC is taken. It was repeatedly washed with phosphate buffer solution and centrifuged to get a supernatant. It was preserved in Elsevier solution for stability. RBCs are resuspended in phosphate buffer solution and 100 microliter of the sample is taken for analysis. The virus along with RBC was inoculated in a 96 well plate and the presence of dengue virus is confirmed by agglutination reaction. Further, heme agglutination inhibition test was performed in the plant extract in order to find out the efficacy of the plant towards inhibiting the agglutination reaction. For this, drug control was done without virus, cell control was done only with RBC, and test was done along with plant extract and virus together. The resultant is indicating whether the plant extract is having an inhibitory effect towards agglutination reaction. So after confirming the presence of virus in the sample and detecting the extent of inhibition of agglutination with the plant extract, the virus sample concentration was fixed up by performing tissue culture infectious dosage 50 studies. The drug concentration was later done by using MTT assay. This will fix the concentration of non-toxic dosage of the drug used and the viral concentration required for in vitro studies. For TCID50, 1 ml of virus along with 9 ml of minimum essential medium without fetal cough serum is taken, and this was done in S1. Further, serial dilution technique was adopted till S5 tube. From the tube labeled as S3, 200 microliter of sample was taken along with 1.8 ml of minimum essential medium. This was labeled as 10 power 1. 10 such tubes were taken and performed serial dilution. After which, the samples were transferred in a 96 well plate and incubated for 3 days at 37 degrees Celsius for the observation of cytopathic effect. Later, by using Carber's formula, TCID value was calculated. For MTT assay, 100 to 1000 micrograms per ml sample was taken with 100 microliters of MTT. The plates were incubated at 37 degrees Celsius for 4 hours and the supernatant was aspirated and 200 microliter of DMSO was added to each well to dissolve the formosome crystals. It was later observed at 620 nanometer and percentage cell viability was calculated. Further, induction on the morphological change of the cell lines was studied and the docking studies were performed to find out its binding efficacy. From the results obtained at the end of hemagglutination and hemagglutination inhibition and RNA extraction studies, it clearly reveals the presence of virus in the sample. PCR results were compared with the known protein marker found in lane 1 and dengue 4 serotype corresponding to 300 to 400 kilobase pairs. Hemagglutination inhibition test also shows inhibitory nature of the plants towards agglutination reaction caused by dengue 4 virus. Further, cell viability test revealed that among the solvents selected, ethanol showed better concentration limit, exhibiting a maximum non-toxic dosage limit of up to 500 micrograms per ml, and the rest of the solvents were observed with toxicity within 200 to 250 micrograms ranges. Further, in vitro cell cytotoxic studies of ethanol extract showed a normal spindle shape of Vero cells in its first 24 hours, whereas syncytia or rounding up of cells were highly pronounced in its 48 and 72 hours of incubation phase. In order to further prove the efficacy of drug towards inhibiting the CPE caused by virus in the cell lines, the virus along with the plant extracts and the cell lines were loaded in a 96 well plate, incubated further at 5% carbon dioxide concentration for 3 days and analyzed for the effect. From the above image, it is evident that the one which is marked as image A, which is expressed as Vero cell control, wherein monolayer cells appearing congested and haplocyte is seen. In the image marked in B, where the cells along with dengue 4 virus, there are some arrows which inhibits a typical syncytia giant cell formation resulting from the fusion of cells infected with dengue 4 absorbing cytopathic effect. And then in image C, where the cells along with dengue 4 virus and then plant extract were loaded, and it was done with performance of incubation, and the resultant indicating absence of cytopathic effect, the monolayer sheets of cells appearing to be absolutely normal. Further, this in vitro work was justified by an in silico model by adopting a iGemDoc tool, whereas the plants, after performing the characterization using GCMS, the biologically active compounds were further docked and analyzed for its fitness score. From the results, it is quite evident that all the 9 plants taken for the study exhibited a very good binding potential against a methyltransferase domain of RNA polymerase taken for the study, thereby further stopping viral transcription phase. Thank you so much.
Hello everyone. Today I will be presenting on my work on the topic titled Vitex Nigundo ethanol extract protects human corneal epithelial cells against ultraviolet C-induced oxidative stress. The ocular surface is composed of cornea, conjunctiva, and the tear glands. So whenever there is a corneal injury due to UV damage or any other stress, there is a production of ROS which lead to imbalance of the antioxidant enzyme levels, further leading to the production of inflammatory cytokines, which will lead to inflammation and causing oxidative stress-induced cell death. So these medicinal plants, being a source of bioactive compounds, have a major role in protecting this UV-induced oxidative stress. And Vitex nigundo is one among the plants which is known to have reported to have antioxidant, anti-inflammatory potential. It is also reported to have cardioprotective, hepatoprotective, and neuroprotective function. But there is a lacunae in research whether these plants can be used therapeutically or prophylactically in ocular surface diseases. So in our study, we will be evaluating the beneficial effect of this Vitex nigundo ethanol extract in protecting human corneal epithelial cells, where UVC is used as an oxidative stress. So the aim of the study is to investigate the protective effect of Vitex nigundo ethanol extract in UVC-induced oxidative stress in human corneal epithelial cells. Coming to the methodology, human corneal epithelial cells is cultured in DMEM ham-cephal media, supplemented in 10% FPS in humidified conditions. The cell viability assay was assessed by trypan blue assay, both after VNE extract and UVC treated cells. Then the reactive oxygen species level was detected by DCFTA method. The Western blot analysis for corneal epithelial markers such as cytokeratin 12 and 3 and antioxidant enzyme levels for hemoxygenase, catalase, and superoxide dismutase was tested, and the inflammatory cytokine levels such as IL-1 beta, IL-6, and TNF alpha in corneal epithelial cell was assessed. Coming to the results and discussion, the cell viability of VNE extract on UVC irradiated cells was checked, and we found out that the VNE extract was effective in protecting human corneal epithelials from the UVC-induced stress. The ROS level was checked after the VNE extract treatment, and we found out that the UVC-induced ROS levels was reduced after the VNE extract treatment. Similar studies was reported in various other literature, that is generation of ROS leading to oxidative stress is been a cause of pathogenesis of various eye diseases such as photokeratitis, pterygia, age-related macular degeneration, and is also attributed to causing cellular injury, which will increase the membrane permeability, leading to the lipid peroxidation, leading to apoptosis. The effect of VNE extract and UVC was checked on the expression of the corneal epithelial markers, and it was found that the UVC-induced reduction in the corneal epithelial marker expression has been augmented by after VNE extract treatment, and cytokeratin 3 and cytokeratin 2 helps in maintaining the integrity of corneal epithelial cells. A similar study has shown that Central Asiatica extract did not alter the expression of corneal epithelial markers in rabbit corneal epithelial cells. The effect of VNE extract and UVC induced stress was checked on the antioxidant enzyme levels in human corneal epithelial cells, and we found out that upon VNE extract treatment, there was increase in the levels of the antioxidant enzymes such as superoxide dismutase, catalase, and hemoxygenase, and upon UVC induced stress, there was reduction in the antioxidant enzyme levels, which was compensated after VNE extract treatment, which shows that the antioxidant enzyme defense mechanism after VNE extract treatments had led to the decrease in the ROS levels. Similar studies have shown that in rabbit corneal epithelial cells, the hemoxygenase enzyme SO2 was upregulated after hemoglobin toxicity, and the overexpression of this catalase enzyme has known to confer defense against ROS-induced injury, which leads to toxic radicals in corneal epithelial cells. The expression of inflammatory cytokines was checked in VNE extract treated and UVC induced treated cells in human corneal epithelial cells. And we found out that UV and we found out that after VNE extract treatment, there was a reduction in the inflammatory cytokines, and which has led to the protective action of this VNE extract in corneal epithelial cells. UV exposure to cornea causes significant modification in the cytokine levels during acute and chronic inflammatory condition. There is always in previous reports stating that the release of inflammatory cytokines such as interleukins and TNF alpha led to ocular inflammation and uveitis. These are the
I would like to present in the topic impact of Suddhi detoxification. In Siddha medicine preparation, it is a unique mentioned in Siddha literatures. There is a vast science behind the detoxification or purification methods, particularly for Schedule E drugs. Detoxification or purification of your drug involves application of various mediums to interact with the drug and converts its chemical, physical or biological nature of the drug in such a way that the toxic constituent of the drug reduces and the efficacy increases. Schedule E drugs are classified in the Drugs and Cosmetics Act. They are the poisonous drugs, plant derivatives in Siddha system of medicine, but they are used in various ailments. Semicarpus anacardium nuts are one of the drugs listed in Schedule E drugs, and hence it has to be purified before it is implicated dermatitis. Allergic symptoms may be due to the application or ingestion of Assay nuts without purification. Assay nuts are commonly used in Siddha to treat various chronic diseases like autoimmune diseases like rheumatoid arthritis, skin diseases, respiratory diseases, and cancer, etc. With text. to present in the topic impact of Suddhi detoxification in Siddha formulation Serangotide name and its safety data through analytical method ICP-OES. My abstract ID is 206. Detoxification or Suddhi plays a vital role in Siddha medicine preparation. It is a unique process mentioned in Siddha literatures. There is a vast science behind the detoxification or purification methods, particularly for Schedule E drugs. Detoxification or purification of a drug involves application of various mediums to interact with the drug and converts its chemical, physical or biological nature of the drug in such a way that the toxic constituent of the drug reduces and the efficacy increases. Schedule E drugs are classified in the Drugs and Cosmetics Act. They are the poisonous drugs, plant derivatives in Siddha system of medicine, but they are used in various ailments. Semicorpus anacardium nuts are one of the drug listed in Schedule E drugs, and hence it has to be purified before it is implicated in the medicine preparation, because the Semicorpus anacardium may cause some allergic symptoms, particularly it is called as urushiol-induced contact dermatitis. Allergic symptoms may be caused due to the application or ingestion of Assay nuts without purification. Assay nuts are commonly used in Siddha to treat various chronic diseases like autoimmune diseases like rheumatoid arthritis, skin diseases, respiratory diseases, and cancer, etc. Assay nuts have been purified with tamarind leaf decoction of Butea monosperma flowers, cow dung mixed with water and aloe vera juice as per the Siddha classical text. Then Serangotide Nei is a Serangotide or a Semicorpus anacardium based preparation, and it is a popular Siddha drug used in the treatment of lung infections including tuberculosis, rheumatoid arthritis, degenerative osteoarthritis, cancer, skin disease, and neurological pain. Semicorpus anacardium nut was boiled in this preparation. The Assay nuts were boiled in cow's milk and cow's ghee, and hence it may also be called as milk extract of the Semicorpus anacardium. There are vast studies done in Serangotide Nei. It has been proved for its protective role in aflatoxin B induced hepatocellular carcinoma, and in chronic myeloid leukemia mice model, anti-cancer activity of Serangotide Nei has been proved, and it is a promising drug for rheumatoid arthritis, and it has anti-cancer activity. In this study, we have done the elemental analysis and heavy metal analysis of Serangotide Nei and compared with the raw sample of the Serangotide or Semicorpus anacardium nuts. These are the ingredients we have used for the purification of the Semicorpus nuts. Then this is the elements we have selected, and we have done the elemental analysis, because these elemental impurities may cause many toxic symptoms, because they are the renal toxic, they may produce renal toxicity, or aluminium, it crosses the blood-brain barrier and enters into the fetus and causes its toxicity. Then arsenic is a genotoxic and a human carcinogen. Hence, these elements have to be analyzed before the medicine has been given to the patients. Hence, in this study, we have done the analysis for these 8 elements. And the results showed a vast difference in the Serangotide Nei. That is, most of the and all the 8 elements were analyzed, and their limits were below detectable limit only. but in the raw sample of Semicorpus anacardium, the lead was in greater amount, 185.93. And after purification also, it has some lead contents up to 83.87. And but in the processed Semicorpus anacardium based serangotine, it has come under BDL. Hence, like this aluminium, barium, and strontium have been analyzed. They have been mentioned. They have been got a very good result as below detectable limit. From this results, we can have a conclusion that from the raw sample, during the process of purification, that mediums we have used for the purification process may interact with the elemental properties and the physical or chemical nature of the serangotine, and it reduces the toxic elements. Also, the serangotine process, that preparation also helps to reduce these elemental impurities. Hence, the finished product has been given as a good safe medicine. Then in discussion, we can say that I am in the guidelines, ICH guidelines for the elemental impurities. These heavy metals were classified under class 1, and barium and molybdenum under class 3, and aluminum and zinc under other elemental impurities. These elemental impurities do not provide any therapeutic benefit to the patient, and their levels in the drug product should be controlled within the acceptable limits. Hence, we can conclude with the results we have got, the Siddhi process, that is detoxification process, and the medicine preparation process, the steps during the process of serangotine preparation helps to remove the toxic elemental impurities in the finished medicine serangotine. Hence, it is a safe and it is a pure serangotine or serangotine based medicine for consumption by the patients. These are references, and thank you.
Hello, a warm welcome to all. Myself, Dr. M. Anandakrishnan, doing MD Siddha in National Institute of Siddha, Chennai. I am hereby to present the study, a comparative, qualitative, and quantitative study of Siddhar Vaipumuri Sudan and commercial Sudan. Commercial Sudan means camphor. My abstract ID is 210. Let's see about the introduction. Vaipumurai is known as the synthetic preparations. Vaipumurai is synthetic preparation is nowadays is synthetic preparation is doing in industries in large scale. But even though in the olden days, Siddhars like Bogar, Konganavar mentioned elaborately about Vaipumurai, which is known as synthetic preparation of raw drugs like Veeram, Puram, Abin, etc. According to TV Sambasivam Pillai Dictionary, Vaipumurai is described as the synthetic preparation of raw drugs according to the rules prescribed in Siddha science. Nowadays, synthetic preparations are widely used by various industries and commercially marketed. But in the ancient times, Siddhars created Vaipumurai to acquire the purest form of raw drugs in order to avoid scarcity of raw drugs and to ensure the efficacy of the medicines. Sudan, camphor is one of the raw drugs which is commonly used in Siddha formulations. Hence, Vaipumurai Sudan is prepared and is going to compared with the commercial camphor doing the analysis methods. Let's see about the aim and objectives. The main aim of the study is to compare the qualitative and quantitative analysis of Siddhar Vaipumurai Sudan and commercial Sudan. First step is to prepare the Vaipumurai Sudan, to study the qualitative and quantitative analysis, to observe the characterization analysis through SEM, EDOX, and FTIR. Let's see about the materials and methods and preparation of the Vaipusudan. For preparing Vaipusudan, first Pooniru, which is known as Fuller's earth, Vediyuppu, which is known as potassium nitrate, Karvapatai, which is known as Chinnamam Veram. Take these ingredients in equal quantity. Pooniru and Vediyuppu is mixed with water and kept for 4 days. After that, the solution is filtered and boiled till all the water content becomes reduced. Sedimental salt should placed to the another vessel and kept in sunlight for drying. Kuritailam is prepared. Kuritailam means oil extract is prepared from the Karvapatai, and the thylam is mixed with the above prepared salt. Then it is placed in a mud vessel and closed with the lid and sealed with a 5 layer of clay smeared cloth. Then inhumate the mud vessel in ground for 15 days. After that, the horse tongue is put over the inhumated area and allowed to dry for 7 days. On the 8th day, burn the dried horse tongue and allow it to cool itself. Then take the mud vessel out and collect the prepared Sudan. Let's see about what are the procedures followed through these methods. Let's see about results and discussions. In the qualitative analysis, the physical appearance of both the Vaipusudan and commercial camphor are analyzed. The color and nature. The color and nature of the samples are slightly different, in which Vaipusudan means VS has a blackish white and crystalline nature. The sample commercial camphor has a white and translucent crystal nature. Loss on drying. Loss on drying is calculated upon the moisture content of the drug. The loss on drying value for Vaipusudan is 8.93 and for commercial camphor is 9.93. The Vaipusudan shows less moisture content compared to the commercial camphor, which denotes Vaipusudan as a better stability and shelf life period. Then see about the pH. Vaipusudan has a pH of 6.90 and commercial camphor has a pH of 6.80. Both shows the presence of weak acidic nature. Let's see about the quantitative analysis. In SEM analysis is scanning electron microscopic analysis. The average particle size of Vaipusudan ranges from 2 to 3 micrometer. The average particle size of commercial camphor ranges from 3 to 5 micrometer. In energy dispersive X-ray analysis technique, the Vaipusudan has a higher concentration of oxygen and carbon atoms when compared to the commercial camphor. In Fourier transmission infrared spectroscopic method, the Vaipusudan has the presence of primary amines. The line of research work and further elaborate studies are needed to ascertain the therapeutic efficacy of the Vaipumurisudan. First, then next, I conclude that research should be done like this to evaluate the scientific wisdom behind this traditional system. Studies should be done like this. Then I thank the committee for making this wonderful opportunity to present me in this. Thank you.
Good day for one and all. This is Dr. N. Meenakshi, Associate Professor in the Department of Biochemistry, Bharati Women's College, Chennai. I am here to present my research findings on the beneficial effect of gallic acid nanoparticles in descaling the hepatotoxicity induced by sodium fluoride in goldfish. As we all are aware, fluoride is an essential trace element for the normal physiological function of the living system. And it also has diverse applications, not only in physiology, but in industrial preparation as well as in medical science. And though it is essential for the functioning, when it exceeds the threshold limits, it leads to deleterious effect, which is termed as fluorosis. The major way by which it is going to enter into the water bodies is through the weathering of rocks. And once it increases the threshold, it is going to pose a threat on the aquatic species by disturbing the characteristic feature of the habitat. And to have a survival, these aquatic species will bring about cellular changes, biochemical changes, and adaptational behaviors. This fluoride is noxious because it is going to produce free radicals such as superoxide anions, hydrogen peroxide, peroxynitrils. In turn, this will damage the biomolecules in the cell, leading to health hazards. Therefore, survival becomes questionable. Normally, these free radical induced oxidative stress is counterbalanced by the antioxidant system, and this can be supplemented as well. The herbal formulations and natural components are preferred rather than synthetic components to reduce the deleterious effects. And one such compound is gallic acid because it is a polyphenol and it has got wide applications in Ayurvedic preparations. But using it in a native form has got certain restrictions because of its unpleasant flavor, the sparingly soluble entity, and the decreased bioavailability. So the efficacy can be improvised by converting them into a nanoparticle where the size is reduced to less than 100 nanometers. Nano delivery system forms a bridge between the physical and the biological systems. Thereby, all the restrictions posed, such as penetration capacity, uptake capacity, delivery of sparingly soluble substances, bioavailability, and the degradation can be monitored. In the present study, chitosan, which is a polysaccharide, was deacylated and it was polymerized with tripol phosphate by ionic gelation method so that it was converted to a nanoparticle. Gallic acid was encapsulated into it to increase the therapeutic efficacy. The prepared gallic acid loaded chitosan nanoparticles were characterized by FTIR and SEM. The main acquisitive of the present study was done with the fish as a model system. Fish was chosen because it has got the capacity to concentrate metals and minerals. And here in this case, fluoride gets accumulated in the bones, teeth, and also the scales of the fish. And this is an excellent biomarker because it is going to indicate the alterations in the characteristic of the aquatic habitat. Goldfish was chosen because of its inexpensive, easy handling nature and its attractive color. So fish was divided into 4 groups. One was kept as control. The other group was treated with the sodium fluoride, which is toxicant. The other was treated with the gallic acid loaded chitosan nanoparticles, and the fourth group was with the sodium fluoride and the gallic acid nanoparticles, which was co-administered. There was a co-administration. So this fish was these conditions were kept for 96 hours, and finally liver was taken up from these samples. Liver was chosen because it is a crucial organ. It has diverse metabolic activity and it is a major organ of detoxification. So after 96 hours of experimentation, this liver was subjected to biochemical assay of hepatic marker enzymes and histopathological examination to see if there is any change in the liver. Goldfish weighing 50 to 70 grams were procured from the local market, and in each group we had 6 numbers of fish. Standard protocols were followed for the preparation of gallic acid loaded chitosan nanoparticles. It was characterized by FTIR and SEM. The marker enzymes such as ALT and AST were done with standard protocols, and histopathology was studied with eosin hematoxylin staining. Statistical analysis. Student t-test was used to ascertain the significance of variations, and the data were expressed as mean plus or minus standard deviation of 6 fish per group, and the differences were considered significant at p less than 0.05, 0.01, and 0.001. Among the marker enzymes, transaminases were chosen because they regulate both the carbohydrate as well as the protein metabolism. Among the two, ALT is more specific indicator of liver inflammation than AST because of the variations in the distribution of the enzymes in the cellular compartments. The hepatotoxic effect of sodium fluoride is well documented by the elevated levels of alanine transaminase as well as aspartate transaminase in group 3, which was treated with sodium fluoride when compared to the control. And the hepatoprotective effect of gallic acid loaded chitosan nanoparticles is revealed from the reduction in the levels of ALT and AST in group 4, which was co-administered with sodium fluoride and gallic acid nanoparticle, when compared to group 3. Gallic acid loaded chitosan nanoparticle treated groups, that is group 2, it mimics the control, highlighting the intact liver tissue. This was further supported by the histopathological studies where plate A shows a control with the normal architecture of liver with the central vein, portal tracts, hepatocytes and the lobular architecture. Plate B shows the group which was treated with gallic acid loaded chitosan nanoparticles, where you could see occasional inflammatory cell infiltrates and infiltrated RBC in the central veins. In plate C, which was treated with sodium fluoride, you could see areas of necrosis, mild fibrosis, pigment-laden macrophages. And in plate D, where it was co-administered both with sodium fluoride and the nanoparticles, we could see the absence of necrosis and fibrosis, but still there was hemorrhage and pigment-laden macrophages. This shows gallic acid has shown a hepatoprotective effect, thereby the necrosis and fibrosis has been reverted. To summarize, the deleterious effects of fluoride as a hepatotoxin is well documented by an increase in the activity of ALT and AST, supported by histological changes in the liver. The efficacy of gallic acid is enhanced by converting it into a nanoparticle, and that was confirmed by characterization using FTIR and SEM, which is not discussed in this presentation. Administration of these nanoparticles reverse the scenario, which deciphers the impact of polyphenols to combat and quench the stress induced by fluoride. The non-toxic nature of the prepared nanoparticle is also highlighted by insignificant variation between group 2 and group 1. To conclude, the nanoparticle formulations could act as an effective candidate for the encapsulation of bioactive compounds such as gallic acid, a promising alternative to improve their bioavailability for therapeutic interventions. I would like to acknowledge Tanshi for funding the student mini project for PG student, which was helpful to carry out this research work. And I would also place on record my sincere thanks to the organizers for giving me an opportunity to present in this forum. Thank you one and all.